This is TWIS. This Week in Science, episode number 971, recorded on Wednesday, May 22nd, 2024. Something in this show is shocking. I'm Dr. Kiki, and tonight on the show, we will fill your head with stress, hope, and fun. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. The climate is changing. Longtime listeners may have heard about as much as they possibly could care to about the multidiscipline research studies, the multitude of connecting data points, the record-breaking temperatures, the dire predictions, warnings, and alarms being sounded. Some might recall the transition of talking about global warming to defining it as climate change, and the unsuccessful attempted rebranding of climate change to chlamydia. But for anyone who has had enough of it, unfortunately, there is more to come. Because while the past few decades have illustrated the source, the scale, the scope of the problem, what comes next is the age of consequences. And like any consequences, seen or unforeseen, there's always a desire to avoid them. And a regret for not having done so sooner. Be that as it may, you can't unring a bell. The time for action has flown the coop, as all the climate chickens are now coming home to roost. Whenever facing consequences, it is important to keep your chin up, your feet on the ground, and your ears peeled for the sound of This Week in Science coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good. It's to Kiki and Blair.
And a good science to you, too, Justin and Blair.
Hey!
Blair's back. We got
How do I
Blair
run
back in
this
town. You don't have to run it. You just have to be here, talk about the things you love.
Perfect. That I
Share.
can do.
I know. That's what we're all looking forward to. Bring your animal side. Oh my goodness. And everyone out there, welcome to this episode of This Week in Science. We are so glad that you are here, and we hope that you will enjoy this episode. It's full of science. It's been a while since we've had a Blair on the show. A few months, random sightings here and there throughout the world, but we take what we can get. Okay, now, this show is filled with lots of stuff. I brought stories about, what did I bring? The sun, as it is, a promising drug, because those are great, bird brains remembering things, because, well, they kind of do, and something in the brain that makes mice monogamous. That could be a song. What do you have for us? Wait. Justin?
Oh. What do I have? Orange Rivers?
No.
Something that nomads and traditional herders and camel drivers all have in common. And possibly a preprint study that has some implications for how often, maybe, humans and Neanderthals got together, about how long ago, genetic study. And the elephants of Chile.
Chili elephants? Those were mastodons. Wait. Sounds like these are stories that could also be in Blair's Animal Corner. What did you bring, Blair?
Oh, I brought some falcons having fun, and I brought some statically charged caterpillars.
I hope they don't like fly through the air like spiders on their silk umbrellas. So,
No, they just kind of flail.
oh, that's even better. Okay,
Yeah.
flailing staticky caterpillars. All right. We're excited. We must keep this show going and not just be excited that Blair is here on the show because we have a show to do. All right, everybody. If you are just joining us, this is your first episode, or if it's, you've been with us for a while, but for whatever reason, you never actually like subscribed to anything, just want to let you know that we are here doing this podcast broadcast live Wednesdays, 8 p.m. Pacific time on YouTube, Facebook, and Twitch. And those streams get cut into a podcast, and you can access that most places that podcasts are found. Look for This Week in Science, but if that's a lot, TWIS.org is where you can go, find links, show notes, all the things, and, maybe find ways to get in touch with us. But here it is, time for the science, yeah?
Oh, yeah.
Good. Oh, yeah. Well, let's start with that burning, just incredible, hot thing that we can't look at, but we do look at. And it's wonderful and it's life-giving and there's all sorts of, really, the sun is something that could make a person very conflicted if you really think about it. But we have been staring at the sun as humans for a very long time, most recently during the solar eclipse, but NASA and other partners have, we launched and we've had for many years orbiting the sun, the Solar Observatory. And so the SDO, the Solar Dynamics Observatory, has been used, and the data that has been taken by its multiple cameras in multiple color, light, energy wavelengths have been broadcast back to here on Earth. Researchers have used them to try and figure out more and more and more about how to predict solar activity, what's going on in the sun. Why does it have these coronal mass ejections on a fairly regular schedule, like, the 12-year solar, like, higher energy schedule? What's going on there? How can we predict it? Do we need to predict it? Yes, because with satellites and electrical grids that we haven't fixed up yet and all that kind of stuff. But we also want to look at the sun because the corona, when solar flares occur, the corona opens up and there's a hole that gets formed. And that opening of the corona, which is the outer layer of it's actually one of the coolest areas of the sun. The hole opens up, and as the hole opens up, it allows the magnetic field of the sun to escape. And if the magnetic field escapes in the right way, it comes back in a place not too close to where it started. Solar flare, energy from the sun, explosion. We were dealing with that recently. It was a very pretty show for a lot of. It was a pretty show for a lot of people. We really liked it.
Yeah,
and
when you said dealing with it, I always picture when we get the big solar flare, then all the grid will go down. This has always been the thing because we were, at one point unprepared for it, at least.
As we talked about last week, 30 years ago, Sweden had the electrical grid impacted and they went down, and then a Eurovision was occurring in Sweden this year, and I was afraid that Eurovision would go down, but it didn't. So Sweden upgraded. That's what planning is.
Yeah, the biggest thing I heard was whoa, maybe not a good day for racing pigeons. That was
anything really, yeah.
That was the biggest concern this time around. It's well, we were gonna have a pigeon race, and we decided to hold off for a week just in case they couldn't find their way home. Because
now we know that pigeons are navigating based on magnetic information that can be influenced by some of these ejections from the sun and the energy and the way it impacts our atmosphere. But that's also an interesting point because researchers just publishing in Nature Communications have been looking at the waves that occur on the surface of the sun. So when the corona hole appears, there are waves that start moving about. And it's like a sound wave or a focused light wave. And the way the energy bounces around, it can be detected. And this is the first time that researchers are saying that they've looked at this energy in a way that they've been able to see the plasma or the really excited energy, where it's that, not, solid, liquid, gas, plasma kind of thing, where it's this other state of matter. The plasma that is in the corona and how it moves and the waves that move through it, it experiences, or the waves that move through it are kind of like the way that sound can be focused and bounced off a wall. And sometimes you can be in a particular spot and hear echoes or hear someone from across the room as if they're right next to you, or the way that light can be focused within a telescope or a microscope to be used. And in this particular case, they're also looking at if we can understand the way that the waves of plasma on the surface of the sun are interacting with each other and how they get focused through this kind of like bouncing off each other things. Maybe when there are solar flares that are interacting with our atmosphere, that we can potentially understand how our atmosphere is, which is a distributor as opposed to a focuser. Maybe we can take the energy that's coming from the sun and opposed to distributing it, all the energy all over our planet in a way that we don't necessarily like, but maybe focus it somewhere else. So maybe we can turn these focused plasma waves into like a billiards game in the solar system. Anyway, that's me extrapolating, but I'm saying
Wait. To turn it into a billiards game. All of the science is researchers in there are going to figure out a new way to play pool?
Yeah, with energy. So really, it's these waves that we see, what they call magnetohydrodynamic wave fronts. And so when a coronal hole occurs and the magnetosphere comes out and all the waves from that start interacting with each other, then you have a situation that might be predictable. So this predictability could also influence the way that we predict how plasma interacts within some of the nuclear, I can speak, fusion fusion devices like tokamaks, giant tokamaks, and be able to use the dynamics of plasma within magnetic fields and predict it in a better way. And then, on a much bigger scale, be able to imagine how we can take advantage of these things for protecting the earth, for just understanding the sun, understanding other stars, and additionally, are there ways that we can use it to help with our own energy systems? Because what they explain it as is these are dynamics, very physically similar. It's once the hole opens and the ripples start happening, it's like any other kind of wave, and it's predictable and it can be focused, and there is a focal point. And so once we understand those dynamics, it can be very beneficial for our understanding of some of this really not well understood space stuff.
I don't know, they get very vague at the end of this. We've discovered this thing that will allow us to do some space stuff.
Yes.
Leave it there. We'll leave it there for now.
Well, it's not.
Space stuff with it.
Yeah. But this is the first time that we've ever identified this kind of focal point by a transmitted wave within the sun. And so been able to actually, with the Solar Dynamics Observatory and the data it's collected, been able to identify what's happening there and how it was reflected, bounced back, and where the focal point was. It's interesting because it's, the dynamics of it make it not just a simple triangle or, simple geometric situation, but it's very complex. And additionally, This is data that's from 2011. We have all sorts of data similar to this that has not been yet looked at in this kind of way. So maybe there are all sorts of other things that we can look at.
It's very much like the first back of the napkin version of the Death Star. Yeah. If you could have a ripple effect and transmitted wave to a focal point, why, you actually, we'll look at this a little bit. We'll do some space stuff with it.
Don't tell anybody that.
Well, we got some space stuff we can do with this later.
We could harness this space stuff to our benefit.
Yeah, hopefully no evil billionaires are listening to the show.
No, nope. Evil billionaires are
Just
not
the good
allowed.
Just the good
Yeah,
ones.
no. We ban them immediately. All right, who's up next? Justin, you want to tell us about orange rivers? What are they? What is it? Full of tang? Sun-kissed? What's going
This is
on?
Dozens. More than
No.
that. Well, it's everything's once you're into the dozens. Some 75 locations across northern Alaska. Some of the most remote streams and rivers that we have in the contiguous United States are turning from a crystal clear blue to a cloudy and rust-colored orangish colored weirdness.
Algae?
And it could all be thanks to global warming.
Oh,
Yay, thank you.
And yeah, it's not algae. It's not life finding a way here. Researchers from the National Park Service, U.S. Geological Survey, and University of California, Davis, as well as other institutions, sampled these 75, sampled many of the 75 locations. This is quoting lead author John O'Donnell of the study published in Communications, Earth and Environment. The more we flew around, we started noticing more and more orange rivers and streams. There are certain sites that look almost like a milky orange juice. Those orange streams can be problematic both in terms of being toxic, but might also prevent migration of fish to spawning areas.
Because nobody wants a sherbet stream.
It's
Nope.
not.
That's what it looks
not
like.
the
It's not.
preference.
No, it's not tang. It's melted sherbet. It's not okay.
So they started grabbing water samples, trying to helicopter out to some of these remote places that they had found. This is the quote from Brett Paulin, an assistant professor of environmental toxicology at UC Davis, who was a principal investigator in research. The stained rivers are so big we can see them from space. These have to be stained a lot to pick them up from space. So this is a side note story here. Things being visible from space is not as impressive as it may have once been. So it's just
Really? What?
It's
yeah, what
often
camera
used-
are you using, right?
Yeah, it's often used as like somebody's looking out a window of a rocket ship in space, taking a picture with a disposable camera. Oh, I can see this from space. But today you can zoom into like the size of a pizza box, right? You might not be able to read the pizza box, but you could definitely zoom in and see it's a pizza box. So describing something as big because it can be seen from space no longer holds up. Seen from space can actually be used to say how small something is. You can actually, I wrote an SOS message on my roof because I was in trouble. Yes, but you spelled it out with refrigerator magnets, which is so small you can't even see it from space. So,
Well, I'm sure that governments can, but anyway, that's beyond.
and then
Spy
maybe it's
satellites are totally different. But
okay. So maybe they can get down to a license plate or something. I think
Yeah,
that
we
might
can
still
check.
be Hollywood, but that's fine. Seeing things as large as rivers changing color from space by today's
that
standards,
works.
relatively easy.
Yeah.
But that is beside the point. The story was about why are the rivers turning orange?
Yeah. Why, Justin?
Not just that they are. Yeah.
So Paulin, whose expertise is in water chemistry, thought the staining looked similar to something that you can see when acid mine drainage stuff happens. So there's no mines that are near any of these impacted rivers, but it looks very much like abandoned mines. So in abandoned mines, you have a lot of deeply exposed earth and elements that haven't been in contact with oxygen and water. And so you get these leachings of acids and all this other stuff into the water. So they took a look, and yeah, it's kind of what it looks like. Some of the samples from the impaired waters have a pH of 2.3 compared to normal average pH of the rivers, which is around 8.
Which is, it's a little bit less. 7.4, 7.6 is what human body kind of, or regular meat. Like 8 is eight's a little bit more basic.
Yeah, and it's pretty basic, but what they're finding is 2.3.
No, I know that's it. It'll eat your skin.
Super acidic, right?
You don't want that hydrochloric acid.
So,
it's going to eat you.
So what does this have to do with climate change, Justin?
So they think why this is happening is because permafrost is melting away.
Of course.
And as the permafrost is melting away, it's exposing a whole bunch of metal ores and minerals that have been locked up.
Oh, right. This is in Alaska. That's right.
In Alaska, right. So now they're getting exposed to water and oxygen for the first time, releasing acids and metals. There is elevated or high levels of iron, zinc, nickel, copper, cadmium, and the dissolved iron, they think, is likely what is causing color change. Is the large amounts of sort of a combination of acids and iron that are now being released. So part of the problem is, you know where this is? This is in some of the famed Salmon River and other federally protected waters. So, yeah.
Where does all water lead?
This water leads to the ocean.
Rivers run
already
to the sea.
Before that, how many tributaries did they lead into? And are there any reservoirs?
Yeah, watersheds are so important. Yeah.
And in fact, in seeing things from space, satellite images have turned up stained waters now because they went back to see like how big this is, because now it's in rivers. So yeah, they've backtracked it to all these tributaries, which is a great point. They can see those in the satellites now going back to 2008. So they had to kind of reverse and go, oh, what if we looked back further in time? And so this has been a thing that's been growing and growing over time. So anyway.
Do they talk about what they're doing to fix it for it temporarily, trying to block off the rivers, prevent them from feeding into certain areas?
No, because it's rural Alaska, and it's also so many tributaries.
75,
yeah. It's the age of consequences, not the age of solutions. So.
Yeah.
At the same time, it also gives more information about what's happening, and perhaps there are solutions that can come about thanks to technology and changes in the way that we fish and that we use land and water to be able to adapt to the new reality, right?
Yeah, so the fish won't be a problem because they're going to just not be alive. The people.
So couldn't you just dump a bunch of baking soda in there? I mean, it's very acidic, right? So just put all the baking soda in there.
The local people who just will have to find something other than
find something else to do
and water for sustenance.
So find something else to eat and drink. You should be fine. Right?
Come on.
Yeah. Mm-hmm.
Yeah, so I'm looking at one of the last two sentences of this paper. Our initial findings indicate that orange streams are an indicator of impaired water quality and are associated with declines in stream biodiversity and habitat degradation.
Mm-hmm.
So that's it. Somebody do something. That's where we're at.
Nobody's going to do anything. Unless they're like, oh, hey, wait, how come there's so much lithium in these rivers that in our sample? Then they'll be like, let's go mine it for electric car batteries, and then we're saving the future. Yeah, just stop.
Blair, I'm so stressed out right now.
Yeah,
I feel like I'm observing all this stuff, and I want to do something.
Well, let me stick you in an MRI.
Okay, I like that.
And then
no, I don't.
I'm going to have you watch inequality happen.
No.
And because you're stressed, you might act differently than someone who's not stressed.
We have such different weekend habits.
This is a study looking at the cognitive effort related to aiding a bystander versus punishing an offender. And so to better understand that, they put 52 people in an MRI scanner where they then watch someone decide how to distribute an endowment of cash between themselves and another person who had to passively accept the proposal no matter what it was. The participant then decided whether to take money away from the first character who spread out the money unevenly and unfairly, or give money to the second to try to balance it out. And so what they did to simulate stress was they had half of them submerge their hands in ice water for 3 minutes right before starting the task, which is pretty long.
That's a different type of stress than it's physical. It becomes a little psychological, but then you're like, I can't feel my hands.
So I think it's just supposed to be shutting down some of your higher cognitive function. It's just supposed to be stealing brain function away, right?
Right.
And so what their findings found was the
foundings find.
Punishing others requires more deliberation, cognitive control, and reliance on calculations than simply helping someone in need. So this actually aligns with a growing body of evidence that suggests that stressed individuals act more cooperatively and generously. I feel like this has not been my experience as a human, but
I'm the opposite of this completely.
So the claim is that they devote more cognitive resources towards helping a victim rather than punishing an offender. So in that dichotomy, I could see that. In a stressful emergent situation, do you yell at somebody or do you go to the person who's injured or in need of assistance, right?
I yell at somebody while I am going to the the person who's in need.
I think about like when I worked with kids for so long, if one kid hit another kid, you don't punish the hitter first.
You go to
the kid who's crying because they just got smacked, and you can't tend to their needs, and then you punish the kid who hit him, right? So I think that's where this is going. And that's the dichotomy that's really important. You have to have both. You have to have a victim and somebody deserving of punishment. I think if you don't have this very specific dichotomy, then some of this falls apart, right? Like, so this whole idea
a lot of it falls apart.
Stressed individuals act cooperatively. No, I gotta tell you, no.
After living in Portland during all of the protests and the stuff that came from that, I'm going to say people under stress, it depends on the kind of stress. It depends on the situation. It depends on their psychological profile and the position in a hierarchy that they perceive themselves to be in.
Right.
There's a lot.
Yeah. This is deep.
Yeah. And this study, while yes, I think it does verify that even though you're stressed, you can still be good. Yeah. You're still going to help. Like during an emergency, like an earthquake or a fire, you're going to help people who are hurt as opposed to necessarily go after people who are looting and robbing.
I think it says that if you know somebody's watching,
I knew you were going to say that. I knew
your
it.
responses. You will take the most virtuous path you can through.
Yeah. That's the other experiment. Piece is that like punishment is stigmatized. So, are people not supporting the idea of punishment because they're being watched? Sure, that could definitely be part of it. Yeah, I don't know. Yeah, more study is needed if this is something we need to know. I don't even.
I think it's interesting, though, that they say that this, large body of evidence suggests that, people are more likely to do this. And this adds to that body of evidence. Well, great. But let's put that body of evidence together with all these other very nuanced, complex human psychological aspects.
I know. When I was first was checking this study out, I had the assumption that this was like rats or something. Like, oh no, these are straight up humans. Humans did this. Okay. I don't know.
They weren't rats in a cage. No.
Yeah. Anyway, so don't punish. Instead, come to someone's aid.
Oh, so fast. It's so fascinating. How are you feeling? Are you stressed out? Oh, yeah, stressed out, but I'm going to go help that person.
Yeah.
What are these calculations?
There's also some weird aspect of the social engineering. This is like, as long as we keep the population under stress, they'll cooperate with each other. No, I'm pretty sure that's not how anything works.
That's how society collapses.
I mean, and honestly, like, okay, you didn't have to put their hands in cold water or ice water for 3 minutes when you're putting them in an MRI.
Yeah, I was
MRIs are loud and stressful to begin with.
Yeah.
And also, Blair, I'm going to leave you out of this because you haven't been a mom long enough. I'm sorry.
Oh.
I'm still not in the club.
Wow.
But Kiki, here's the analogy I was thinking.
Man, gatekeeping already, huh?
Always. Kiki, here's how
join us. No, you're not one of us.
Have you ever perhaps yelled at your child in a way that you wouldn't have done in public? In private, like, hey, you're not doing anything, in a way that you wouldn't have done if you were like out in public place.
I cannot remember the answer to this question.
Oh no! Like I will be have my Papa loud voice. Like
there's a loud voice, but then there's the like,
hey, knock that off.
Yeah.
What are you doing? Hey, come on, like that kind of stuff, right? Which, if I was in public, I would never do that because I would be like, I don't want to give the impression like this is how I always talk to my child. I want to, you know, does that make sense?
Mm-hmm.
I don't want people to be like, we should call somebody. But at home, I'm going to definitely use the Papa indoor loud voice to get that message across. Is it just me? Maybe I'm exposing like, okay, I'm a terrible dad. I didn't even realize.
You're not a terrible dad. I think all parents come to a moment of stress and overload where they find themselves publicly doing something they never thought that they would. Like, what? What am I doing there? And, at a certain point in life, some people have brain changes that are degenerative and that change the way they would normally react. And so it's not even really the way they would have responded in history, or, things change about how their brain works. And so Alzheimer's disease is a very common cause of dementia. We've talked about it a lot on the show and various hypotheses like the beta amyloid plaque hypothesis, which may or may not be part of what we know amyloid plaques build up, but what's leading to that? How does it, how can we really create treatments that are going to positively influence Alzheimer's patients and how their disease progresses? Some researchers years back, they started thinking about it differently. And they're like, well, maybe we don't attack the plaques or other things, but what if we support the nerve cells? And what if we find ways to target the cells to help them be healthy and happy? And then maybe because they're doing great, the plaques won't progress as much, and maybe they'll do okay. So this new drug, there's a clinical trial study that was just published in Nature Medicine. And this drug is called LM11A-31, developed by Dr. Frank Longo and UCSF professor Dr. Stephen Massa. Frank Longo is from Stanford. It targets a particular receptor in neurons that's called P75, and it's a neurotrophin that regulates survival. So instead of making a cell die, it says, I'm going to be young and healthy forever, which is great. And so it basically promotes the signals that contribute to growth and happiness and all sorts of stuff. Anyway, so they've got this drug going on, and they were like, oh, okay, we really got to do a clinical trial. And basically, the first clinical trial you do is, let's do a clinical trial that isn't, that just shows that it doesn't kill people. Is this drug going to be safe and not do bad things to people? So they did that, and they ended up with what they call phase 2A clinical trial data. And they took that data, which they sent, and they saw that the drug was tolerated, no real bad side effects to anybody. And they sent it to some researchers at Schulich Medicine and Dentistry, the School of Medicine and Dentistry, to do positron emission tomography and look at samples of cerebral spinal fluid from these people. And they didn't really know what was going to come out of it. They were just, nothing bad, right? Let's just make sure that there are no bad signals. There's nothing happening in the immune system. Nothing's weird. And they found that it really did change the metabolic and the immune signals that would normally be found in an Alzheimer's suffering person. So in this study that they published, it was like 300 some odd people in the study, and they've got all of them that tolerated the drug really well. And they were mild to moderate Alzheimer's patients, so not anyone who had progressed too far. But in the study, they saw that it seemed to be like not allowing the disease to progress as fast as it normally would. And in animals, they had previously shown that it preserved neurons and reversed damage to neurons. And so it was really exciting to actually see this P75 target, this drug targeting it, actually impacting. They saw changes in inflammation. They saw a whole bunch of results that were really, really positive. 2 synaptic biomarkers specifically that they were looking at were very positively enhanced. And so this is, clinical trials. They go on, and you have to get through them all. But this is not an animal trial. This is not just the results of a do we not hurt people trial. This is getting to the point of actually coming up with beneficial results that could possibly allow people who have been diagnosed with Alzheimer's to not degenerate with the disease as quickly, and possibly to even survive longer. And if there are other drugs that come along in the meantime, perhaps the next treatment's around the corner that could really get rid of whatever's causing the disorder.
Yeah, some of the clinical trials, each round of the clinical trial has, I think, the most success rate of somewhere below 30%.
Mm-hmm.
So even if you've made it through 2, then there's still 30%. And then if you made it to 3, there's still like a 30%. Like it falls off very quickly, the number of things that go into this pipeline that make it out the other side. However, we are in an age where drugs are incredibly more targeted, where the molecule of the drug has a specific target, as opposed to trying a thing that seemed to have an effect across. We don't know exactly. Past drugs have worked without us understanding the lock and key mechanisms behind why. We just discovered correlations, pursued those, and then kind of got some kind of a result that seemed positive. What we're in the age that we're in now, we are at such an ability to do that lock or see the key and then design lock for it, or see the lock, design the key for it, however the go. And
however
then,
it works.
And then build that and put those into pathways. So even though clinical trials historically have terrible outcomes in terms of the odds, things like this that already know the mechanism upon which they will work and are designed to do just that have a much greater reliance. So if anybody's hearing the stories like this and going, but you guys are talking about clinical trials and, that's too, it's not your grandfather's clinical trial. We've got a lot, I think we got better odds with some of these new targeted medicines.
We do. And I think again, I see this story as promising, but again, it's early. And what it means is that it is going to go into further clinical trials, and we'll see how the results go. But so far, it's working well. And it's the interesting thing
so far it's
is
not
that
killed anybody.
But it's also potentially helping. We're not seeing really bad outcomes. We're not seeing bad immune reactions.
Don't get ahead of your skates there, researchers, in your clinical trial. We saw a good result from a thing that wasn't designed to show good results. That's nice. That's not what your job was. Your job was just not to kill anybody. Congratulations.
Don't kill anybody. You didn't. And there were some interesting results. And so basically, we'll get more clinical trials
to yourselves.
Using
move on to the next.
Using a different treatment philosophy, which I think is a very interesting factor here. All right. Let's talk about another story. You're going to depress me now, aren't you, Justin?
Huh? Yes.
Yeah.
What do camel drivers in the Sahara, nomads on the Mongolian steppe, traditional herders the world over, all rely on most to sustain their livelihood and cultures?
Water
and
Food?
water and rangeland.
Rangeland, yeah, okay.
The wide open spaces, lots of land, and the starry skies above.
Is that a song?
It is when you sing it.
Okay.
Anything's a song when you sing it.
It's true.
Filled with next to nothing, save the wild grasses that the livestock love to munch on, and shrubbery, whatever their herds are munching on. New research into these habitats globally finds that as much as half of all rangelands, and this is everything from Arctic to the tropics, deserts, mountains, whatever you want to call a rangeland, or 50% of them are degraded, meaning they're not producing what they once were able to. Mostly natural grasslands used by livestock and wild animals to graze upon. This also, of course, scrubland, mountain plateaus, desert wetlands. They kind of looked everywhere. This is from the UN Convention to Combat Desertification.
Good luck.
Yeah. It's like top reasons they offer up for why everything looks like it's on the down slope. Climate change, urban expansion, population growth, and the conversion of land for farming were fueling the destruction of open spaces. So, oh wait, so it's pretty much people. And just the upside though is they also discovered that healthy rangelands are actually really good asset in the fight against global warming because they lock away quite a bit of carbon in the soil and spur the growth of vegetation that pulls planet heating CO2 out of the atmosphere. So they also discovered some really good news about the rangelands that they also discovered were being degraded worldwide.
Which is interesting. Like years ago, there were conversations about, how to ecologically sustain various rangelands across the United States where ranchers have their cattle and their sheep and allow them to go, and how cattle can actually degrade watersheds because of the way that they walk into the watershed area to get access to the water, and they disturb that ecosystem in a way that is not beneficial to the ecosystem at large. So there's this real interesting interplay between what we call rangelands. They're called rangelands because we put ranging animals on them that we cultivate. But at the same time, those animals can have negative impacts. But at the same time, then we're like, oh, no, we need corn. And then we're not taking care of the soil and doing proper sustainable practices. And like this, let's just go back to making sure that we save the pandas.
Well, and it's one of those things too, like
save everything.
I thought you were going to say, let's go back to crop rotation. And I was like, yeah.
No, we're going to go further back.
Okay.
So also like we think of rangeland quite often in the West as like cows, right?
That's how I think, yeah.
Cattle farming, that kind of thing. But there are like somewhere in the neighborhood of 500 million people, according to this study, who are pastoralists. 500 million people across 100 different countries, mostly kind of poor and marginalized communities, such as, and this is, I know it's probably too late. Choices have been made in my life already that probably rule this out. But at some point, I always assumed I was going to go and join the Bedouins.
Nope. You didn't do that.
Traveling as like a salt merchant from town to town or something like this.
Maybe in 18 years.
It just felt like, sleeping out underneath the stars of the Sahara. I just felt like that was going to, at some point that was just going to happen.
It could still happen.
This is the rest of my life. I am now a wise old man wandering around with my Bedouin community. But it's a lot of people whose lifestyle and culture has been doing this for years. It's a lot of Africa is open rangeland for the wild animals that we still have on this planet.
There's lots of areas.
There's a whole lot of countries and continents and places where rangeland is supporting quite a large population of humans and animals. So to see that half of it is on the downtick, not good news. But the upside is that we now know that rangeland was capturing quite a bit of carbon before it was degraded.
And what is there still is, and so perhaps we can conserve not just the rangeland, but also the cultural ways of life of the people who support who live in it and what's happening there. There's a whole huge turtles all the way down, everybody.
At some point that we might have to be like, hey, if you want to be a human, you have to live in a city.
If you want to be a human, upload your soul, consciousness, whatever you call it, to 1-800-WHO-CARES. Now, everybody, I would like to thank you for being here with us for this episode of This Week in Science, in which Blair has come back to talk about animals. It's so amazing. Justin's going to have to leave in about 15 minutes, so we're going to keep rolling on. But, if you really, really love this show, I ask you, please, to head over to TWIS.org, where you will find links for our Zazzle store, where you can get merchandise, which also we get a portion of that money, so it helps to support the show. Our Patreon, where you can sign up to help support the show in an ongoing fashion, $10 or more a month, and I will thank you by name at the end of the show. Or just get the links to different episodes. Go see the show notes, see what we did, share the pages, share TWIS.org with your friends. Because the more people who come to enjoy TWIS with us, the better this will all be. Yeah, it's true. Thank you for helping us do what we do with you, for you. Can't do it without you. All right, everybody, let's come on back to some more This Week in Science. Now, I would normally specifically go to Blair's Animal Corner at this point, but because Justin has a hard out, I'm going to switch the little times and stories. And Justin, you got to get going because we got a hard out.
Yeah.
So,
okay.
Yep. It's all you now. Justin's not an Animal Corner.
Okay, I'll go with a quick version. First of all, I'll go jump down to Chile. So it turns out thousands of years ago, early hunter-gatherers returned regularly to a lake called Tagua. It's not Tagua Lake, it's Tagua Lake in Chile. And they went there to hunt elephants.
Makes sense. It's a water source.
Yeah. When I say this was thousands of years ago.
But Chile.
This is in Chile. This is somewhere around 12,500 years ago. Very long time ago. Notably, the site features the fossil remains of also a Gomophorifera. I don't even know what that is. It's an extinct relative of elephants, anyway. So there's signs of butchery on bones, along with stone tools and other evidence that indicates
signs of butchery on that name. Shoot.
Yep. I wasn't gonna say it.
I can't pronounce. I don't know. Gomphothera? I don't know. It's not one of the ancient creatures that we're super familiar with,
or that you care about a lot.
I can't pronounce. They also found fossil cactus seeds, bird eggshell, suggesting that a camp was occupied specifically during a dry season when they would do their elephant hunting. So, wow, 12,500 years ago, which is, what is that? Is that, right around the Younger Dryas. So maybe this was a new behavior to combat, like maybe humans had to go after the big prey during Younger Dryas, or maybe it caused some sort of conflict. Anyway, it doesn't matter.
Younger Dryas was warmer or colder?
So it was a big climate shift that had different effects, different places.
Right. Okay.
Yeah, depending on where you were.
I made it too simple. Yeah.
Yeah. Okay. Last story of the night, then I got to roll.
No, wait, what?
What?
You're done already? That's it? I thought we were going to talk more about the elephants.
Yeah. We could talk. I said it was going to be a quick one, but go for it.
Yeah, okay. But you can finish it up. But why are these really what's important about these?
Did you know there were elephants being hunted 12,500 years ago?
I knew.
Chile?
Are these related to mammoths? Is this I don't think of elephants as being North or South America. I think of mammoths maybe. But
there were mammoths in
this
North
is different.
America for sure.
Yeah.
I'm not really sure what was going on. I can't pronounce the thing. Yeah, Gomophorifera, whatever it was.
Gomphotheriidae.
There you go.
There you go with it. Yeah.
Wait, did you go back? Can you go back to that slide? This is a part of the story that I didn't even catch.
This table? Yes, there's a table.
They found a horse?
Horse. What? Yes, what? There were rodents, Artiodactyla, birds, amphibia, reptilia, actinoturgii.
Okay, so Gomphotheres, or whatever you want to call them, are in the Proboscidae order, which includes
mammoths,
elephants, all of the current elephants that exist, but also, yes, mastodons.
Mastodons, okay.
And mammoths.
Oh.
So they're all
this
related.
Is a okay. But this is a particular species that they're looking at.
Mm-hmm. Yeah. They have big,
but it was suitable
looking tusks. They're funny.
And so people were like, "Hey, look at this lake. It's awesome. And these big animals come here. We can eat them seasonally and survive." And that's part of the way it goes. Mm-hmm. Is that what's going on?
Yeah. It is just I guess I'm just always wanting to point out that 7,000. This is 7,500 years before current modern Europeans were in current modern Europe.
Yeah,
there was people in South America hunting elephants.
Yeah, and so there's the 13,000 year ago evidence that's North America, blah, blah, and that's been pushed to 25,000 years ago, and so 12,000 years ago in Chile that's a whole nother thing, which is very exciting. Yeah. All right, next story. What do you want to say?
Next story. Birds do it, bees do it, even ancient humans and Neanderthals did it, but when, where, and how often? Analysis of ancient and modern genomes suggests contemporary peoples' Neanderthal DNA mostly came from a single prolonged period of mixing around 47,000 years ago. The findings suggest that during the early days of this gene flow, wherever it occurred, because they're not exactly sure where, roughly 5% of the interbreeding population's genes came from Neanderthals. This is meaning that approximately 1 in 20 individuals in the population that ended up being ancestral to all of the currently less African descended individuals was a Neanderthal. 1 in 20 of the ancestors of the early days of this gene flow. It's kind of hard to
As much as we've all had offspring over the generations, that seems like it was a large percentage that would have an influence.
Well, so it's like if you are if you had one of those dating apps and you're going like through 1 out of the 20 of them would be a straight up Neanderthal. I guess is what the dating scene would have looked like.
Man, 1 out of 20. That sounds better than my online dating career.
Oh my gosh.
Perfect. So this is one of the couple of the things is this is a preprint. So I think this is going to have to get
peer reviewed.
It's not
commented
just peer reviewed. It's going to have to get, there's going to be follow-up analysis to, I think, this, because it's actually they've used as many ancient humans as they can and done basically a backtracking of Neanderthal DNA and segments to see, to sort of pinpoint the time when they were, when these coupling events took place. And so the further back you go, the longer and more kind of complete these Neanderthal DNA segments are. And by sort of seeing how they have changed over time, that allows them to sort of do a reverse time thing. They use genomes, ancient modern human genomes from around 275 present day individuals from around the group. So that's that's not a huge sample. I don't know why they've only got 275 present day individuals. That to me is kind of weird. Like, it seems like you could use more, but maybe you just don't need it. Because really what you're likely seeing is a lot of repeats, but as long as they've got a good global sample with that 275, it's probably okay. Some of the contentious stuff, though, is if there's this coupling event at 47,000 years, and yet we have these same segments that are in people in Australia, it could indicate that maybe Australia didn't get settled as far back as we think.
Yeah.
But it could also mean that there were earlier populations that had earlier different Neanderthal segments that either dropped them or that those populations came to an end. Some of the ancient humans that they looked at actually have this. They have somewhat longer, different Neanderthal DNA ancestry, but those are not reflected in current modern humans. So that might mean that particular lineage didn't progress. So there may have been bottlenecks that sort of limit our view into the past here. There's also some sort of There's also it's kind of interesting that the very oldest genomes for modern humans held these long stretches, but in just a few thousand years after that, the Neanderthal segments were much shorter. It sort of suggests that the current modern humans were kind of getting rid of some of the early long stretches of Neanderthal DNA, which may have been because they carried maybe deleterious mutations or led to infertile offspring or something of this nature. So if there was stuff that didn't work, we may have gotten rid of it. And it also confirmed that some modern humans acquired some things that involved in skin pigmentation, immune response, metabolism that we ended up hanging on to.
Yeah, I love the idea. I mean, if you just think of it as like, oh, yep, Neanderthals and then humans met and then ta-da, then we have Neanderthal DNA. It's like, wait, hold on. Let's think about how that would work. Yes, Blair, with your it's the braided stream, right? I mean, humans had the braided stream. And then, it's all these people going all over the place, and you have tribes and you have people coming in and coming out and like entering into relationships and leaving relationships and offspring, and those offspring go and do their own thing. And there are, in the early days of humanity and other hominid species, there's so many, there were not many people, right? We're not many, hominid people. I'll say people in the very anthropomorphic kind of way. And so when you found people, no matter who you were, you'd stick with them. You'd have a tribe if they let you in, right?
Well, that and also
right?
Despite being,
and you'd go wherever they went. So,
despite being speciated to some extent across time, humans have been using tools for millions of years. So as we've evolved technology more than we have really had to adapt our biology to new environments. It happens to some extent, but not to the extent you would see in most other wildlife, which means we've been able to intermix with other versions, more disparate version cousins than a lot of animals might have had success with, because our evolution tends to be an adaptation of behavior, less so than a physical adaptation to environment, if that makes sense.
And there are a lot of interesting philosophical and also mechanistic questions that go along with it that a lot of people are studying right now, which is kind of cool. But Justin, I think you're about to hit your out time, and you know what time it is right now?
Is it time for Blair's Animal Corner with Blair?
She loves our creatures, great and small. Biped, milliped, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
It's been so long.
Oh my goodness. I have playful falcons. I just love stories that are like, hey, this animal plays. Hey, this animal over here, it plays. Hey, did you know that this animal, it plays? You'll never guess. Well, there's another animal that plays. It's the striated caracara that is from the Falkland Islands. And I guess they have kind of a bad rap there. They're these large, dark birds. They're falcons. They attack smaller birds.
Yeah, they eat babies. Yes.
They don't. They do. Well, okay,
bird
yes,
babies.
not human babies. Sorry,
bird babies.
They
Yes,
eat bird babies. You're correct. And
mouse babies and all the things. Yes.
So so they're seen as kind of a nuisance and scary. So part of this is like, hey, this scary bird. It's not scary. They play, but also let's get into what they actually found, which is just anecdotally they saw that these guys would play around with plastic, sea cabbage, sheep poop, stones, and it didn't really seem to have a rhyme or a reason. It really looked like they were playing with it. So then they did some observations. This is Messerli Research Institute at the University of Veterinary Medicine in Vienna. They did a bunch of observations of these guys over 3 years, and they found numerous instances of what they considered play. A play behavior needs to meet the following criteria, which we've talked about on the show before, but just to kind of remind everyone what our criteria is. it needs to be intrinsically rewarding. So they're not getting anything else out of it. There's a lack of apparent purpose. This generates new behaviors or experiences. It's not something they would do otherwise. It's voluntary and repeatedly performed, and it occurs in relaxed conditions. They're not under stress. They didn't just get their hand out of an ice bucket, right? And so some of the notable events included 3 young birds catching and releasing a dried bundle of kelp roots, 2 others playing tug of war with penguin skin.
Oh boy.
And they didn't eat it. And another bird picking up a piece of
like
sheep
a dolphin,
dropping it, watching it roll down the hill, retrieving it, and doing that 6 more times. So
playing
yes, it's
fetch.
like a dolphin or a toddler, or yes, absolutely. And so this is play. And they started doing tests to figure out what makes objects attractive for them to play with. And so in the case of humans and other animals, the researchers have looked at this with, usually their attention is towards more complex things. But that's not true, because how many times do you buy some fancy toy and they only just want to play with the box, be it a cat or a dog or a human child.
Cat, child.
They want to play with the
same.
box. It's just like that. So they had an array of objects varying in complexity, and a cone was the least complex. And you guessed it, that cone, they loved it. That was their favorite toy. And so this is a systematic investigation of play in wild animals, and they're going to continue for one reason, to be able to anticipate how they will interact with objects, to try to improve relations with these birds, because they are threatened. And so if they know what will keep their attention, how they interact with new objects, and whether their play attracts their peers, that can help minimize the risk of toxic bait being an issue for them, there being human conflict directly with the birds, and it can just help them generally conserve the species. So there is a conservation benefit from all of this, but also it is just cool to see yet another species engaging in what we call play.
I love this so much, but I think one of the things that we talk about a lot is what is the adaptive benefit of play, right? And we talk about, play in developmental periods is it's beneficial for learning, because you try different things out and it allows you to learn different stuff, and you are trying different scenarios, imagining different things. So is this something that is only a younger bird behavior, or is it something that is all ages? If it's all ages, like, humans, we play video games. Lots of people play games, all kinds of games late into their life, and it keeps life fresh. There's a stress relieving aspect of it. There's a social aspect to it. Parrots, very similar as a social species. Play is important for that. So what is their social structure? So how are these caracaras socially integrated, and why would it be important?
Great question. It seems like there were a lot of young birds involved, but it doesn't appear to be exclusively young birds. But you're right. I don't know very much about caracaras social dynamics. Be interesting to see, because usually birds of prey, specifically falcons, are kind of monogamous, not really communal animals. So I would expect that this wouldn't be that different. But the fact that they had a trio of birds playing with things,
and a trio of birds playing with things, and then the one that just played fetch with itself and a ball of sheep poop.
A ball of sheep poop. It's sheep poop, everyone's favorite toy.
No, just that one. Falcons. It's awesome.
Yes. Kiki, you want to hear about statically charged caterpillars?
Oh, yeah. I'm ready. What do I have to do to have them really attached to my sweater?
Yeah, good
but
question.
First, Justin, you just sat down. What are you doing? Where are you going? How is it going? Is everything okay? You're muted. Right.
I gotta go. But I have to hear this story.
Oh, okay. The short version is, and then I'll get into it.
Highlight.
Their statoelectric fields are carried by numerous animals, including some predators like wasps. And caterpillars with those fuzzy bodies can sense the electrostatic fields and respond to the presence of predators flying through the air. So they'll curl up or flail around. And the way they figured this out was by catching a bunch of wasps, measuring the static electric field around the wasps, and then emitting those and other similar but not the same fields around caterpillars in a lab. And they responded to almost every static electric field, but the one that was most similar to the wasp electrostatic field, they flipped out.
This is fascinating. Wow.
Yeah, so they can respond to an artificially generated electrostatic field that matched what is created by a wasp in nature.
That's wild.
Yes.
It is.
So talk about a sixth sense, right?
Yeah.
So like this, you can sense something going on around you. You're not feeling the wind. You're not hearing the wasp. You're not smelling the wasp. There's no other sense involved. You're just sensing the static electricity coming off of their wings with your little furry body.
And it's making you all tingly.
Yeah. It's a bad tingle. It's a I'm gonna get eaten tingle.
So it's going to be their wings interacting with the air in those molecules, but then also like the little hairs on their body and so much.
Sure. Yep. Yeah, so we know about static electricity as a balloon against a rug, or when you scratch your socks across the floor. But prior research has shown that many animals emit an electrostatic charge as well. Flies, spiders, bees. And then similar animals are also able to sense them. So they kind of put it all together and said, if wasps emit it, what gets eaten by wasps? Caterpillars, they're covered in those fun hairs. Let's figure it out. So they captured 200 caterpillars of 3 types. The cinnabar moth, the vapor moth, or scarce vapor moth, and the European peacock butterfly. And
I liked the morth. I really wanted it to be a morth.
I haven't done this in a while.
It's okay.
Very much mouth.
It's fine. It's good.
But yeah, let's call it a morth. Why not? So some flailed bodies, other coiled into a defensive posture. And so they really can sense when there's a predator coming just by the electrostatic charge coming off of them.
So David Hudd in the chat room is saying spiders have hairy bodies. Do they also have these spidey senses?
Yes,
there's like all kinds of fuzzy bugs, right?
Yeah.
There's all kinds of hairy little fuzzy things. And you're just thinking like, well, that's an unfortunate look. You just that happens to be why bugs look that way. They just, they're hairy and fuzzy for no reason whatsoever other than to look weird, right?
Hey,
I mean, that's obviously how I think about everything.
Did Stan Lee know about this and not tell anybody? Because this is the best explanation of spider sense I have ever heard.
Oh my gosh, it is.
Yeah, totally. But then they would have to draw that into the character too. Aside from having all these, also got like.
I think there were frames that had like the hair rising. It was Spidey sensey.
Yeah, it's the waves around his body. It's totally an electrostatic field. I buy it.
Totally,
it's perfect.
Stanley was ahead of everybody on everything. Mm-hmm. So
it was a magneto electrostatic field.
Is this whole world of this communication going on in Bugland that we're just?
Oh yeah.
Assumed. Didn't assume was occurring?
We assumed all sorts of mammals were mute because we couldn't hear them, but they were just talking lower than we could hear. We assumed plants don't talk. They're clicking at each other. There's all sorts of communication in nature that we just don't have the tools to sense. But they have a whole conversation going on.
Now I want to test and see if bearded men can...
It's not during November.
You have all these hairs on your arms that stand on end when things happen.
So now we need to take cyclists and swimmers who, use depilatories and get rid of their hair for their biking and their swimming competition so they can be faster and more aerodynamic and see whether or not their sensory engagement with the environment changes when they take their hair off. We have people we could use for these experiments.
Yeah. Right.
Already.
I think we're numb to it. I think we.
Oh gosh, I forgot I had to go.
You gotta go to work.
Bye, Great to see you again, Blair.
Don't miss your train. Bye. Oh my goodness. Okay, so we've got these awesome caterpillars, which caterpillars are the early version of moths.
The larval stage of a moth or a butterfly.
Thank you very much.
You're welcome.
Yeah. So they like eating normally like plants and stuff, and gardeners get mad at them all over the place. But I mean, really, so wasps are attacking them and eating them and injecting them with their eggs and
yeah.
And so I never actually thought about the fact that a larval moth or butterfly might have defenses other than the ones that are toxic.
Right.
That if when you touch them, you get an itch or a rash or, they have physical defenses, not this sensory defense. So what do they do with it? Do we know they just sense it? We don't know what they do with it.
Curl up and hide.
That's it.
So I guess if you're a caterpillar on a leaf and you sense a wasp coming and you flail around like this, you probably fall off of the leaf onto the ground and therefore avoid getting swooped down upon, would be my guess. Is that it's a Hail Mary. It's like, I'd rather fall to my death than get eaten by a wasp.
Because there's always the chance that if you fall, you can crawl.
Yes.
And then you might, with your senses, but be able to...
you'll get to hide amongst the leaves down there and stuff.
Yeah.
Might be enough to get away.
Wow. So they basically were like, here's a wasp electric field and we'll take it away and we'll see what you do. And the caterpillars were like, yeah.
Oh no.
I got it. But it does seem to be, now the next question is, right, how does it happen? Are the electrosensory structures the hairs like we're talking about?
Right? That's the assumption, but yeah, you're right. We haven't not figured that out yet.
We thought that pigeons and other migratory birds or homing birds, first it's like, oh, it's little otolith structures. Oh, it's little physical things. And now it's like quantum physics.
Right.
Yeah.
No, that's fair. Obviously the next step is to shave a caterpillar. No, I'm joking. That would not work. I think actually what would be next would be to breed caterpillars to lack the hair. Is what you would need to do.
That would maybe be easier.
Yeah.
Oh my gosh. What'd you do for your graduate degree? Well, I shaved caterpillars. It's one of my favorites. Can't wait to see that dance your PhD. That could be great. Yeah. Oh my goodness. This is a really, really cool one. And of course, they gave caterpillars, full consent forms to fill out ahead of time.
Oh, sure. Yeah.
And anyway. I love this. I'm never going to look at caterpillars the same way again.
Good. Those woolly bears.
Woolly bears. Honestly, I've, always just looked at them going, oh my gosh, they go from this state to being moths or, butterflies or whatever they become. But never.
And they become a soup in between that we have recently learned is not fully homogenized.
Right.
It is disorganized soup.
But it's not chaos.
No.
Yeah. I love it. We're learning all sorts of things. Well, when I was in grad school, I studied bird brains and
you did.
Yeah, I did. Lots of experience with bird brains. And I was involved in studying a species that we call the western scrub jay in the San Joaquin Valley of California. One of the authors of this new study that has come out in PLOS One is my previous PI, my mentor for many years, Nicola Clayton. The lead author on this, however, is James Davies. And in this study, they are not looking at scrub jays in California, however. They are looking at Eurasian jays, which are just these beautiful blue-colored jays, but they're not mean like the Steller's jays. Like Steller's jays are these, if you've met them, they're the ones that are like, and they get mean and they're mad at you and all this kind of stuff. These Eurasian jays, they got their own thing going on, but they're also social, very much jay-like in the way that they exist within the environment, and also cache food that they have to remember where it is, so it helps their survival, so that it's a very strong evolutionary force for these birds, and because they're social, and also the need for this spatial memory has led them to where we're looking at them now. When I was looking at the scrub jays, we gave them experiments where it was like, okay, we'll give you a tray and it's got all sorts of weird shapes on it, colors and shapes and everything. And so it was at a certain time of day on a certain day of the week. And then we'd give them dog food kibble that would last basically for as long as dog food kibble lasted. And then we'd be like, oh, okay, no, but here is another tray with a different set of shapes. But this one has wax moth larvae in it. Wax moth larvae degrade really quickly, like over 24 hours. Like it's like bad food. They're going to get food poisoning, don't want to eat it anymore. And so we tested whether or not the birds could remember the time and the context of the shapes and the colors and all the things that were involved in what they were remembering. And at the time, the results suggested that the western scrub jays had what we would call an episodic-like memory, where with people, we call it episodic memory because we can all go like, oh my gosh, something triggers it. And you're like, oh, this morning when I was at home and I put my keys in the bowl and and you remember all these details, and you remember the details, not necessarily because you're expecting to have to remember the details. You're not planning on having to remember the details. It's just something that your brain does. If somebody triggers you to remember it, you can remember all the things. And all of a sudden you're like, I don't know how I know this, but I know this thing. But I remember the whole day. I remember that car over there and that noise and that thing. And you can explain an entire episode at a particular time, place, and all the stuff in it. So the work that we did with the scrub jays, people said, well, is it really episodic memory or episodic-like memory? Because maybe those scrub jays were planning, they learned that they were in this situation where they were going to get the same trays again, and they just had to learn to remember stuff so that they could get the thing that they wanted in the future. So maybe what they weren't really doing is just, really episodic human-like remembering. But maybe it's more like planning for future events, because we had to train them on this thing and they had to learn how to do it. And so in this study, what they did with the Eurasian jays is they gave them a completely different scenario in which they varied the situation in a way that wasn't related in any way to planning. And what they call it is incidental encoding of information, which I think is just a really interesting, it's details that are unimportant, it's incidental, but you remember them. And so this study is the first one that suggests that these birds, these Eurasian jays, have this ability because they put the Eurasian jays, Garrulus glandiaris, into a situation and had them, one of multiple sources of food had food in it that they might like. They had to just be there at the right time, the right place, and look at all the different things. And maybe they didn't have to plan on remembering any of them because they were all so different. But then in the future, they were given these things later in different conformations, and depending on what they paid attention to, they may or may not have been able to identify the place where their preferred food would be. But in the case of these birds, they absolutely did. And they were...
So basically you're saying like, you put out 4 types of food and they each had a label on them. So like this picture that you're showing, my favorite thing is mealworms, and the mealworm was under the square.
Right. And
but there's no
also,
conditioning to know that I should need to remember that.
Right. Why would you? Because maybe the mealworm's under a circle a different time. Maybe it's under a triangle a different time. You don't know that. Or, maybe the color, it just has to do with a particular time and a particular place, and understand. So the big thing is like the different kinds of food is the big determinant, because when we were using decomposable food like mealworms or the waxworms that would go bad really quickly, what we saw with the scrub jays is they would be like, I'm not going to look for that. I don't care where that is. A week later, they were more likely to choose where the dog food was as opposed to the mealworms. But in this particular situation, because the contextual information was different about shape and color and all of the like incidentals, it wasn't as like specific. It wasn't, I took a mealworm and put it under this particular red Lego a week ago, and now I don't care about that. I want the dog food kibble that should still be good that's under the round green Lego. That was very specific. This made it, all the contextual stuff, more difficult for the birds to discern. But when put in the situation, these Eurasian jays were able to find the food they wanted to eat, which suggests that these birds are paying attention to a lot more information, similarly to humans, and probably similarly to a lot more animals that we just haven't tested in this way previously. Yeah.
Yeah, I mean, it's a game of survival, right? You need to remember as much as you can about everything always to stay alive.
Exactly. And it's the question of like, migrating birds. What are they paying attention to? Are they just paying attention to the stars, the sun, the magnetic field? Are they, when they get to where they're going, what are they finding there? I mean, what are the local cues for plants and like the kinds of trees, like the color? Is it coniferous? Is it deciduous? What kind of food sources might there be? Like, there's so many contextual cues that like we think that, or we ignore all the time, or we take for granted most of the time. Yeah.
But we associate things with things that don't exist at all, right? Like,
because our brain likes to.
This was associated with this number. This number is speaking to me. Stuff like that.
No, it's 9:34, not 11:11. Totally. Last study related to brains, because I love a good brain. For years we've been trying to figure out, what makes animals promiscuous or monogamous? Blair, why did we choose a single partner for our kids?
Well, in the animal kingdom, there's no true monogamy, and I could talk for hours about that. But there's a bunch of reasons.
Which is why I've
it usually has to do with the care of the babies, or it could have to do with the care of the mother, or it could have to do with resource availability, territory. There's a lot of things.
You're like, yeah, there are many details that we don't understand. And that's a big part of it. So researchers at Columbia's Zuckerman Institute have just published a study in Nature of their work on 2 species of rodents And these mice are the old field mice, and they're old field mice are monogamous, and they're really, really close relatives. The deer mice, Paramiscus maniculatus, are promiscuous. There are lots of differences between them, though. They're in different environments, they're different sizes, they have different food needs. So there's a lot of stuff behaviorally and ecologically that's different, but they are very, 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 very closely related. So when they're like genetically, look at them, they are closest relatives with each other, first cousins-ish. But deer mice are promiscuous, old field mice, monogamous. What makes the difference? Researchers looking beyond animal behavior and ecology and all that kind of stuff, they're like, yeah, let's look at the brain. And so they looked at the brains of these different rodents, and they discovered there is a unique adrenal cell type within the brains of the monogamous mice. The monogamous mice that have this unique cell type also have a brain region which is within the a cell type. It's in the adrenal gland. It's called the zona inaudita. Inaudita? I don't know if I have the pronunciation correct, but one of the researchers who now works for Regeneron, but was a graduate student at the time of this study, said, oh yeah, no, we called this zona inaudita, which is Latin for previously unheard of zone.
Because
Because
sure.
yeah, no one ever observed this type of cell in another animal. Okay, so the adrenal glands are divided into 3 zones. This particular monogamous mouse has a cell type. They called it a 4th zone. Ooh, new thing, zona inaudita. This is everybody. If you're going someplace you've never heard of, you're like, oh, I'm going to zona inaudita. Wow. The cool thing about these cells, this zone, it makes the adrenal gland of the monogamous mice like 6 times heavier than the adrenal gland of promiscuous mice. They discovered that also these adrenal cells release way more of a hormone called 20-alpha OHP, and this 20-alpha OHP is related to nurturing behavior in mice, which leads to more parental behavior, and is probably related to this linking of 2 mice together to raise their offspring together, whatever. So the big question now is, can they take what they've learned, which is that, okay, this zona inaudita has a gene that's called AKR1C18, that helps create 20-alpha OHP, which is found in humans and other mammals. So is this going to be something that we can look at in humans so that we can use it when we've gone on the dating apps and then make people more monogamous when we want them to be? No.
Or just screen people. How's that gene looking? No, specifically, right, you got to turn it on in the, what was it, the field mouse that's super promiscuous, and then you got to turn it off in the deer mouse that's...
No, yeah, so the old field
mice... Did I do it backwards?
The deer mice are promiscuous, so you need to turn it on. I did it backwards. So they did that. They did that in the mice.
Okay.
And they were like, hey, promiscuous mice, we're going to give you this gene. And they became more monogamous.
There you go.
They didn't act as polygamous. They had fewer mates. They spent more time caring for their young. It was totally out of the ordinary. Mm-hmm. And then when they removed it from the old field monogamous mice, it made them seek out partners more often and not be as monogamous. So what is this going to do? Well, maybe it'll help us understand a little bit more about parental care. And if we try to make too much extrapolation to humans, that's just dumb.
Yeah.
But
no thanks.
Yeah, no thanks. I mean, all the things we do with things that we should really just not do. That's great. But really, postpartum depression might be something that
that's interesting.
That when mothers are depressed and have this feeling to reject their offspring, maybe this is something that could help as opposed to just an antidepressant.
Right.
You know,
a chemical part of couples therapy. No thanks.
Right.
Yeah. That's very interesting. I think that this is the part of evolutionary science that gets really interesting because you have, okay, I know why some animals are monogamous and some animals are polygamous. It's because there was selective pressure towards monogamy. So there was a evolutionary benefit to being more monogamous. But what was the cause of that push, of that variation that allowed for the selective pressure?
Right.
And was it just a random mutation, or was there some sort of hormonal trigger that changed the growth in the brain? Like, that's the other part that gets really interesting is, was it random chance, or was there something that pushed adaptation forward chemically based on what was going on? Like,
right, which is why I mentioned
oxytocin have to do with all of this.
Right. And, you know, as I mentioned before, these mice are, they're closest relatives, but they are different sizes. They have different ecological histories. They have different survival needs. So they have evolved separately for a reason. And so this idea. But is this a cause or an effect? Or none of either. I don't know. I had another story that I think Justin really would have enjoyed, but we're not going to talk about it. Basically, everybody LARPing for science. Researchers in Europe took old armor that was found from the, I can't pronounce this correctly, the Mycenaean era. This armor, they found it in a village, and it goes back to the age of Homer and the Iliad. And so they were like, oh, we've got the Iliad. We've got all this historical stuff that was really stories, whatever. But then they copied the armor and were like, we're going to do it. And they got Marines in Europe to volunteer, and they put them in the armor and made them fight for 11 hours, which is like what Homer said, like an epic battle would be. And so they then tested all of them to be like, how's that fight? How are you feeling? How'd the armor do? And they said, yeah, that was pretty good armor. It was great.
Wow.
Yeah, nobody expected.
Did they have orange slices afterwards?
They should have had orange slices. I don't know. They got water. They got to have a sip of water. I don't know. Had snacks.
Did they have foam swords? What did they have?
They made spears with copper tips that were blunted and not so that they wouldn't actually hurt each other. They made swords. Right. They made arrows. They reproduced this whole thing and then put them in like a temperature controlled laboratory so that it'd be like the temperature of the time and like all this stuff.
They didn't even fight outside. What kind of good is that?
I don't know. It's really, I mean, honestly, Ren Fair's done. It's like we're going back to the age of the Iliad. It's the Iliad, the Homerian fair. I don't know. Everyone's gonna be making this armor now. But there was a question as to whether or not
that's pretty fun.
or not it's really fun. Yeah. everybody's like, oh, I found this armor. Did it really work? Why did they have it? Nobody thought they really were good fighters. Was it really a good thing? What did Homer say? Oh, I don't know. He told stories. And now they're like, well, maybe they actually were fighting and doing a lot of not just politicking, but actually fighting. So
I love that they were like, ooh, we could put this armor on a mannequin and like throw things at the mannequin or, use different pressure sensors. No, I'm going to wear it and we're going to fight.
And it wasn't just volunteers. They got volunteers that matched the height and stature descriptions from the old stories and that would fit in the armor that they found.
Sure.
And that also matched like skeletal height and stuff. So they were like, honestly, they were like, we're reliving this again. Can they fight?
We're going to have a mess around with grant dollars, but we're going to do it right.
Seriously, everybody's going to be making this armor. It's going to be the big thing. Seriously.
Wow.
Mycenaean LARPing. It's happening soon.
I love it. Can't wait.
Okay. We've got you up late and I'm glad you've been here, Blair. Thank you so much.
Yes, it's been lovely.
Been so awesome. And everybody else, thank you for joining us for another This Week in Science. Blair, do you have any final comments for this episode before we go into the shutdown mode?
Thanks for being here, everybody. That's
awesome. all. That's it. That's a good thing to say. I like that. All right, everybody. We are going to head on out. So thank you for being here. I want to say thank you to everybody who is a part of making this show happen. The co-hosts, Justin, who has left on a train, hopefully, that he didn't miss. Blair, thank you for being back here again. Everyone in the chat rooms who are here watching live, thank you for chatting, commenting, being a part of the conversation and part of our community. And for specific people, Fada, thank you so much for all the work you do with show notes and social media. Identity 4, thank you for recording backups of the show and all this stuff. Gord, Arnlore, others who help make sure that the chat rooms are nice places where people are nice to each other. That's really awesome. And additionally, Rachel, thank you for editing the show. And all you Patreon sponsors, thank you so much for all you give to this show. Thank you to Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazarb, John Ratnaswamy, Chris Wozniak, Vegard Shefstad, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan Shubrew, Sarah Forfar, Don Mundus, PIG, Stephen Albaran, Daryl Myshak, Andrew Swanson, Fred S104, Skyluke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp. I hope you don't come across any of those orange rivers anytime soon. Lawn Makes, EO, Adam Mishkan, Aaron Luthan, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, and Patrick Pecoraro and Tony Steele. Thank you so much for everything you have done over the years and for helping this show keep going. If anyone out there wants to add their name to that list, head over to twist.org, click on the Patreon link, and choose your level of support. We will be back again next week. Oh my gosh. So what are we going to be talking about? I don't know. Don't miss it.
Yeah, we'll be back Wednesday at 8 p.m. Pacific time, broadcasting live from Twitch, YouTube, and Facebook.
And if you want to listen to us as a podcast, just make sure you look for This Week in Science wherever podcasts are found. If you enjoyed the show, I hope you did. Get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twist.org. Also, you could sign up for our newsletter.
that'll happen. We love your feedback. If there is a topic that you'd like us to cover or address, suggestion for an interview, please let us know on one of those social media accounts out there, or like actually just send us an email, because I think that's the one thing that works right now. But you got to put TWIS in the subject line, because why, Blair?
Oh, it'll be spam filtered to the Falcon Islands, where a falcon will just pick it up over and over and drop it down a hill, and we'll never get it
because it's like fetch sheep poo. Okay.
Yeah. And we look forward to discussing science with you again next week. If you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. Science is coming your way, so everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science, This Week in Science, This Week in Science, This Week in Science.